How does a spectrophotometer work? A solution that contains the pigment of choice is exposed to different wavelengths of light. The transmitted light strikes a photoelectric tube so that it can convert the light energy into an electric current. A galvanometer is then used to measure this electric current. The solution is tested with each of the colour's wavelengths in order to determine which wavelengths the pigment does and does not absorb. Once all the wavelengths are tested, the data is gathered and used to plot the absorption spectrum of that pigment. The absorption spectrum is a graph that plots light absorbed versus wavelength. Plant leaves have two main types of pigments, called chlorophyll and carotenoids. There are two kinds of chlorophylls, chlorophyll A and chlorophyll B. The chlorophylls play a major role in photosynthesis. The chlorophyll pigment's molecules absorb light in the blue, violet and red wavelengths. They emit a green colour because they do not absorb green light. Chlorophyll A is the chief pigment in photosynthesis. The accessory pigments include chlorophyll B and the carotenoids. Chlorophyll B is slightly different to chlorophyll A. Its role is to funnel the energy it collects to chlorophyll A, which occurs in the light reactions. Carotenoids absorb light in the blue-green region. They are responsible for giving plants their red, orange, yellow hues. Like chlorophyll B, they also funnel their energy to chlorophyll A during the light reactions. Carotenoids have a very important role in plant leaves, called photoprotection. When light strikes a leaf, it hits the leaf with a high concentration of energy. This means that there must be a way for the excess energy to get dissipated and absorbed, without causing damage to the chlorophylls. Carotenoids absorb that extra energy and spread it around, so that it doesn't harm the chlorophylls. Carotenoids also act as antioxidants by reacting with reactive oxygen species that would otherwise cause damage to the cells. When a photon strikes chlorophyll, the energy of that photon is transferred onto an electron. The electron gets excited and moves from ground state to an excited state. When the electron falls back to its ground state, it releases some energy as heat. Sometimes a lower energy photon is released, emitting light. When a pigment releases a photon of light, fluorescence occurs.